The structure of DNA that we see down here on this page is one of the most iconic images in the study of biology. I would even go as far as to say it's one of the most important discoveries in the 20th century, if not the last 500 years. This chapter is about nucleic acids, and when we say that, what we mean is DNA and RNA. And in biochemistry, we're always interested in structure and function. So our first unit goal is to describe the structure of DNA and its close relative RNA. And note, we say we're talking about components, linkages, and that's a great connection to what we just did with sugars and fats, where we also talked about components and linkages. The second goal is to understand how DNA and RNA are synthesized, or replicated, we sometimes say. Now, there's a few things I'd like to talk about in this introductory video that will help us frame the whole thing. The first is a structural issue. We said in our very first unit that proteins and peptides are polymers of amino acids held together by peptide bonds. That's our theme of components and linkages. Nucleic acids are polymers of nucleotides held together by phosphodiester bonds. So as with proteins, we're going to start off with a monomer. We're going to start talking about nucleotides, and then we're going to link them together as we've done many times. We've already talked about phosphodiester bonds. We address those in the lipid chapter where you have two alcohols buried in a phosphate. More on that to come. The second thing I'd like to address in this intro is the function of nucleic acids. And we can do so with something you may have heard in the past, and that is the central dogma of biology. And what that means is the way that information is stored and transmitted in cells, genetic information that is. First of all, we can say that DNA is an information storage molecule. You no doubt know that from high school biology or other classes you've taken. But what is the information in DNA? Tell us, what's it's there for? What is genetic information? Well, that's, as you can imagine, a very complex question. But the upshot of it is, especially for a biochemist, is that the information is the blueprints for making protein. Maybe we should even make a note of that. And as a protein chemist, someone who studies enzymes, I always have a protein-centered view of things. DNA is there to tell the cell how to make protein. And this happens not in an easy step, but in a complex series of steps. The first thing we do is take that information, which is sort of a long-term storage of information, protected in a stable molecule, DNA, in the nucleus, and we make a little copy of it. And I like to think of this as if the DNA is a rare book in a library, protected, kept from harm, and only used occasionally. We go in and make a photocopy of that. That's what I think of as RNA. That's a quick, easy, cheap, disposable version of that information. And the RNA molecule is actually the blueprint for the protein. So let's just make a note of that. This process is going to be one called transcription. Transcription means when you copy something, but you do so in the same language. And DNA and RNA use the information in the same language. They have the same alphabet, nucleotides. You can see that DNA and RNA are very similar in their structure. The next step is to take the information in that temporary photocopy, or the RNA molecule that is, and to translate it into a different language. We're going to now take that information and put it into protein terms, protein language. And it's the order of the nucleotides and the information in there that gives us the order of the amino acids that we've been talking about all along. And of course, eventually, protein folding, once it's synthesized, gives us proteins, and that is where the function of most important things happen in the cell. So that's how information is passed from DNA into protein to make the machines and structural elements of our cells that are so important. Let's back up and talk about how we make DNA.